Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaut Einvoll. One of the main successes of systems neuroscience is the discovery of orientation selective cells in the primary visual cortex called V1. In the early 1960s, Jubel and Wiesel found that cells in this part of cortex respond most strongly to bars and elongated visual stimuli oriented in particular directions. They also explained how these properties stemmed from arrangement of synaptic inputs from cells in the visual thalamus, the brain region placed earlier in the so-called visual pathway. This observation suggested that neurons in the visual pathway are so-called feature detectors, with neurons in higher visual areas, that is, areas higher up in the visual pathway, representing more and more complex visual features. However, when pursuing this program started by Jubel and Wiesel to higher visual areas, the results have been mixed. It has been difficult to identify clear visual feature detectors in neurons in these brain areas, at least as convincing as the bar detectors in V1. Today's guest, Li Zhaoping, has a theory for why this is. Li is a theoretical neuroscientist and has since 2018 been a professor at the University of Tübingen and a group leader at the Max Planck Institute of Biological Cybernetics at the same place. Her idea is that V1 is a saliency detector, which identifies the most interesting, or salient, spot in the visual field. This information is in turn forwarded to the parts of the brain that governs eye movement, which directs our gaze, or central vision, to this salient spot. In contrast, the job of higher visual areas is primarily to analyze in detail what is seen by the central vision. One nice aspect of this theory is that it provides falsifiable predictions, both in terms of physiological experiments, for example the spiking of neurons in V1, and in terms of psychophysical experiments, where human test subjects report on visual observations. In the podcast, we talk about the challenges of vision, in particular the so-called attentional bottleneck, implying that only a small fraction of the information present in V1 can be forwarded to higher visual areas. We further discuss the difference between what Li refers to as unconscious looking and conscious seeing. We next talk about the two main elements of our theory, first the V1 saliency map theory, and next her idea of so-called central-peripheral dichotomy that explains how the higher visual areas perform the seeing. And towards the end, we discuss how her ideas about how vision works might generalize to other senses, like hearing or touching. Note that Li is very active in outreach. She wrote a very nice book, Understanding Vision, Theory, Models, and Data, in 2014, and she has put out a lot of educational material on her webpage, including a set of video lectures on vision. The link to her webpage can be found on the homepage of this episode. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoreticalneurosciencepodcast and choose a membership of your liking, for example at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings, as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast, where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners and hope that all of you can help spread the word about the podcast to other people in the community on social media or otherwise. Okay, here is Li. So, welcome to the podcast, Li Zhaoping.
Thank you. It's a great pleasure to join this adventure.
Yeah, yeah, it is an adventure. So, so you're one of the first podcast guests. Of course, we have, I think I met you first in the 90s or something. So it's been
That
a
was a
while,
long time ago. Yeah.
long time ago. We were both beginners. I was even more a beginner than you, but at least we were sort of at the early
Similar
parts of our,
physics background. Yeah,
yeah.
exactly.
So, So you worked on vision for many
Yeah.
years. You've written many papers and even a book,
Mm-hmm.
Understanding Vision, Theory, Models, and Data, which I really like and recommend. And
Thank
of
you.
course, there will be a link to that on the homepage of this episode. And you also made an online course, even, and you also made a school or something, or a course.
Yeah, it's a System Vision Science Summer School, and this is more or less kind of dictated by our interdisciplinary nature of our
Uh-huh.
community. And so, therefore, you need to teach across disciplines, and that book and the online course is they're both for the purpose of easy communication and learning across disciplines. Yeah.
Excellent. Yeah, yeah, because your kind of vision research is quite cross disciplinary, as we'll come back to. And you are now in Tübingen, right? At
In Tübingen, yeah,
yeah,
Max Planck Institute for Biological Cybernetics, and with the
excellent.
university, yeah.
Yeah, excellent. So, I think sort of, and since this is going to be about vision, I think it's good to start with a story that maybe many of us knows, the story of like the mapping out of receptive fields in the early visual pathway. I
Mm-hmm.
guess like with the Kufler, we found like these circular receptive fields in the retina,
Mm-hmm.
and then Jubel and Wiesel, so they moved on to V1. So
Very
what?
quickly, they
Yeah,
did very well. Within 10 years, they achieved a lot.
yeah. So what did they achieve?
So basically, they're trying to say, okay, you find these retinal neurons respond to spots of light, and these spots, they call them features, and those neurons are therefore feature detectors.
Hmm.
So the natural question is, what is the feature detector in the next stage along the information pathway? And
Hmm.
so they went to V1, which is not exactly the next stage, but after the LGN, which they also worked on, which is the first next stage, and
Hmm.
V1 is the first stage in the visual cortex in the brain,
Because
neocortex.
LGN also had circular receptive fields, right?
That's right. And
Yeah.
so they first went to the LGN and then
Mm.
realized that, well, the feature detectors in LGN roughly they are the same as those in the retina. So they moved
Mm.
on to V1. So they're really productive. Yeah. And so
Mm.
they said, what is the feature? And then they found, okay, now it's not just the feature is one spot of light, but
Mm.
as a little line, a little line segment. You can say one line is made of two dots linked together. So it seemed to be very natural if you think about it, but nevertheless, it wasn't known. So they need to discover it, and they
Mm.
discovered all through this several years through the anatomy, through the physiology, and that was really
Mm.
fantastic.
Yeah. So they map. So this is a story about this like different neurons responding preferentially to bars and maybe moving bars and bars in particular orientations, and so
Exactly.
on. So this is of course the story that everyone likes to tell. But then if they try to move beyond that, going to sort of like higher visual areas like V2,
Mm-hmm.
whatever V4, and I don't know quite sure what all these areas are called. I guess it depends on what kind of animal you're talking about. But
Mm-hmm.
so then it was not so clear cut, or the success was more dubious, or
That's right. So, in a sense that you say, okay, these feature detectors, how big are these features?
mm,
They're actually not big enough to, let's say, image your whole face standing in front of me. So, therefore, if I just was looking into one neuron, I can only see a small peck maybe on the corner of your mouth. So, I would
mm,
not be able to recognize your face.
mm,
Well, then you say maybe the next stage along the information pathway was a bit bigger. Maybe you can see your whole mouth, and the next stage you maybe see your whole eyes.
uh,
And then the feature will get more and more complex, not just
mm,
one spot, not just one small lines, but become
mm,
a huge face. And look at the huge house and a huge
mm,
tree, and that was the hope. And
yeah,
so imagine if you are Huber Weasel, you like to continue to the next stage, you might want to look for it.
mm,
Or if you're not Huber Weasel, you could be another scientist and say, let me try another 10 years, maybe I can find the next stage.
mm,
And however, that seemed to be not as expected from the progress they
mm,
made from
yeah,
retina to V1. You cannot
yeah,
extrapolate the same progress to V1 to
exactly.
V2 and so on. And
Yeah.
so that was quite unexpected.
Yeah, because I guess Jubel and Wiesel went on to study like development and other things
That's right.
because
That's
the
right.
program didn't really sort of work for higher visual areas.
Yeah.
So, and I guess you have this, and it's interesting if you sort of, as you say, when you talk about this in some of your excellent talks, which I've seen before this recording, is that sort of is that there hasn't happened too much in the last 50, I mean, the 50 years after, I mean, you say 1962 maybe, where they found these particular orientation selective fields,
Uh-huh.
and even in 50 years later, they commented themselves that, well, it's the field hasn't really moved so much. So that moved so much. It hasn't been so much progress.
That's right. No, you can say actually the move has moved a lot or not so much, depending on your point of view.
Yeah.
So, what has moved a lot? The recording techniques have moved a lot from single glass pipette to multi electrode and two photon imaging. And these technology have moved a lot. And and a lot of work have put in. You can the field, the number of scientists in the field has grown a lot. The conferences get bigger in terms of
Yeah.
exactly like the expectation from how much it moved from retina to V1, maybe in terms of that, people trying to find the next stage feature detector seem to be really, really difficult. And
Yeah.
yeah, you can find spots of progress, but none of them seem to be at the similar level as
Yeah.
what Hubo and
Yeah.
Weasel achieved on V1. And
At least
that's
it's
just
not
what,
such a
yeah.
clean and beautiful story. When you sort of talk about neuroscience, you often like to talk about these V1 receptive fields, and because it's something that you can explain to a layman, but it's at least
Mm-hmm.
not so clear.
Mm-hmm.
Not so clear. Sort of like, what do I tell the layman about higher visual areas? I
Mm-hmm.
guess you have
Mm-hmm.
these object cells in IT, and like with like the Jennifer Aniston cells and
That's
things like
right. The
yeah,
grandmother cells, people say, can we
yeah,
find a cell that respond to a whole face?
mm,
And recently there have been some progress on that. That's also quite exciting.
mm,
But there's lots of kind of no man's land in between. You don't find
mm,
a cell respond cleanly to, a triangle or a star in a way as clean as a V1
yeah.
cell respond cleanly to a little line segment.
Because one thing I think this is sort of like your idea here is sort of this that what has been missing from this kind of program is this idea of attention
Right.
and this that you have this phenomena of attentional blindness. So what is attentional blindness to you?
Yes, so in a sense that uh, people have shown that you can show people one photo, a second photo. Let's say the one photo you have something in the background. Let's say a tree. A second photo, suddenly the tree is not there. People may not even notice the difference if you put a blank in between two photos. So photo one blank and photo two, it's just such a huge difference. Could not manage. It could people did not notice.
Yeah.
And this is attentional blindness, and it's kind of a discovered only about 20 or 30 years ago. It's really this dramatic demonstration was discovered this way.
Yeah.
Even though technically, when people trying to measure the attentional, bottleneck capacity, it was half a century ago. People knew then we cannot uh, process as much information, such as if I speed up your podcast by 10 times, I couldn't comprehend. Okay, have to slow down a little bit. Nor can I speak 10 times faster. Yeah. And so this bottleneck is also in speech as well as in looking at the photograph in vision. And it's just there. And unfortunately, it wasn't quite connected with how we study the visual system from retina to V1, V2.
Because I guess because one very dramatic and fun sort of demonstration of this is sort of like these basketball players where you're told to sort of count how many times they pass the ball to each other, and then
That's
there's
right.
this gorilla passing in the back that
Uh-huh.
you don't that
You
that
did
you
not
can
notice there's a
no,
gorilla walks through the
yeah,
basketball
yeah,
court because you're engaged with counting the
yeah,
basketball.
exactly. So using your like attention on the counting the basketballs. And
That's
then,
right.
and when, but it's also interesting that after you told about it, you kind of sort of, how could I not see this?
That's
Right?
right.
Yeah.
And that's
So,
right. And so being told is like your attention being guided. When
mm,
somebody point to the gorilla, did you see it? This pointing is to guiding your attention.
mm. So, and then you mentioned this bottleneck that you have, like this idea that you have I mean, that's sort of your key idea, that you have all this information coming into primary visual cortex from the eyes. And I guess we can talk about humans, like, even though it's sort of like similar things will happen with other mammals, at least advanced, like higher mammals. And then, so all this information coming into V1, primary visual
Yeah.
cortex, and then there is this bottleneck that you cannot really take this information. I think you estimate it's like 20 megabytes
20
second.
megabytes is assuming that you have 20 pictures per second. If
Yeah.
you go to movies, it's about 20 pictures per second.
Yeah.
And let's say each picture is 1 megapixels.
Yeah.
Let's say each pixel is 1 byte.
Yeah.
Then it will be 1 picture is 1 megabyte and 20 images 20 megabytes.
Yeah.
However, they're picture related to each other, so they're not exactly non-redundant.
No,
so
but
if you
still.
then compress them, then you can go to 1 megabyte. Roughly 1
So
megabyte each second coming in.
that's
That's a
1
lot.
million bytes.
That's right. So 1 megabyte, you can have a whole thick book
Yeah.
of War and Peace and put it in your disk. That's only 1 megabyte. But you cannot read this whole book in 1 second. Yeah, because you can't read it.
No.
So therefore, you cannot even read 1 page.
Yeah.
And so maybe I can read 2 sentences. So therefore, reading 2 sentences in a huge book of text, this is only text, not images, is how much information is throwing away.
But then you say that, well, there's no way that all this information that is in principle in V1 can be sent to taken up by the higher visual areas. So there is this bottleneck, and you have there are these estimates of sort of like the amount of bits that really can be eventually reach whatever, like
it's about 30 to 40 bits per second. That was estimated in 1950s. That's right.
40 bits per second. That's really not much.
That's not much. It also depend on the point of view. And say, okay, 1 megabyte, that's a whole book. And 40 bits really is about 2 sentences. But nevertheless, you can also say it's a lot. 40 bits is about 10 digits.
Yeah.
So if I tell you my telephone number in 10 digits in 1 second, it's actually quite a lot. So therefore, you can say 2 sentences is like 10 digits.
So of course, this bottleneck is not only in the first stage from V1 to V2. It's from V1 up the whole way. But it's the idea then that sort of like that our vision, that you have this visual representation, that internal visual representation, and that is updated with maximally, say, 40 bits per second. Is that how to think about it?
This is how people have measured. When they first measured it in 1950, people found it surprising, really so low.
Yeah.
So this experiment was actually repeated. And because at that time it was very popular information theory about how much is the channel capacity of a channel. And they say, okay, a channel could be a radio station, a channel could be your telephone wire, but what if a channel is your mental information channel? And so this 40 bits per second was actually repeated by multiple experiments, and people never found anything more than, let's say, 100 bits per second. So order of magnitude, it's repeated across multiple
references. I was actually discussing, I was out sort of hiking in the woods with my friend before this podcast, and then we started discussing how low this is, and we thought, how can you sort of manage like walking on walking in a bumpy track in the forest, a path with a lot of obstacles and stuff. That seems like it just would be hopeless if you only get 40 bits a second of information.
Yeah. But 40 bits per second, as I say, it's 10 digits of numbers.
Yeah, but
if
still.
I tell you, what is my telephone number? It's 5234 1288902.
That's true.
In 1 second, it's actually a lot of information. Because each number, each bit, each digit could be 0, 1, 2, 3, 4, 5, 6, 7. So it could be 10 possible choices. As 10 possible choices, 2 to the 3 is 8, and 2 to the 4 is 16. So therefore, 10 choices, 2 to the 3 point something. So 3 to 4 bits per digit. Yeah.
But anyway, so regarding this particular number, which is frighteningly low, or maybe surprisingly low, but nevertheless, the main idea is that the visual, your brain can only process a small fraction of the information that enters the eyes, and then you have this eye. So then you distinguish between sort of looking and seeing.
That's right.
Yeah.
And
So
so
what is meant by looking in this?
seeing.
It's
So the
not
idea is
synonymous like many people would use
that's
in
right.
maybe regular life. So okay. So what is looking
What
and
is
seeing in
looking
this?
and seeing? So the idea is you have 1 megabyte per second of information coming, but you can only do 40 bits. So therefore you say, I have to select 40 bits out of 1 megabyte. How do you select which 40 bits? Okay. And so selecting which 40 bits is looking. So in a sense, I look at this spot, I'm selecting this 40 bits. Look at
Okay.
that spot, I'm selecting that 40 bits. So the idea is you
So a
look
little bit like focusing, almost like
it was actually more of a selecting. It's in a
yeah,
sense as if
select.
a
Yeah,
big river of data coming in.
yeah,
There's suddenly a small channel only allows
yeah,
40 bits per
yeah.
second passing through.
And you
Then
select.
you can say selecting which 40 bits. And so in a sense, you say, what? The 40 bits must be very important for my survival. Yeah. And so therefore you say, I can
Yeah.
read through the whole book of 1 megabyte and find the most important 40 bits. But then by the time you read through the whole megabyte, you already dead. You see, so in
Yeah.
a sense that you have to go fast. So
Exactly.
therefore, you need to find the most important 40 bits before you even read through the book.
So you need
Yeah.
to find the lion. Yeah.
Yeah. So if the lion is jumping at you, that must
Yeah.
be
Yeah.
within your 40 bits. Otherwise, you'd be
Yeah.
dead. And
Yeah.
so that's the
Exactly.
idea. And so therefore, to select that, it's almost like having an egg without a chicken. So in this sense, this is actually a very, very difficult problem. And
Oh.
so looking, so for instance, I look at you. If I look at you, I cannot tell in my periphery there's something else. So for instance, when you read,
It's a gorilla passing behind me.
well, this is probably most easily demonstrated when you read. If you read
Okay.
a page, when you read the beginning of the line on the left, you
Mm,
cannot read the word at the end of the line to
mm,
the right. Yeah.
mm,
So the thing is, you're looking to the left of the page. You cannot read a word on the right of the page. In
mm,
fact, you cannot even read a word. Let's say 10 characters down the line or 20 characters. You already cannot do it. So
mm.
that's the idea.
Yeah. So
Yeah.
then you have looking, and then you say so that's sort of like selection, and then
Selecting
with
this 40 bits into the
yeah,
bottleneck.
yeah,
Then
and
you say, what is this 40 bits? Is it
yeah,
telling me a lion? Or is it
yeah,
telling me Gauta is there? Is it
yeah,
telling me that my daughter just
yeah,
just
yeah,
just falling asleep? Or
yeah,
something.
yeah, exactly. So that's sort of like the seeing part, and
Seeing.
that's sort of like seeing is sort of when it reaches your like becomes part of your perception.
That's right.
Yeah.
That's right.
Yeah. So, and of course, one thing is so then you have several problems. I'm going to sort of discuss them. But the
Mhm.
first, of course, the problem is that if you find something in this, like in this, while looking, that you select this particular particular area of the visual field that you want to sort of select and investigate in more detail, then you
Mhm.
need to get your central vision
That's
right
right.
to so you need to point because that's one important thing with our vision is that they have this much more receptors and cells dedicated to what's happening in the center of your gaze.
That's right. That's right.
So that is sort of what these saccades do, right? They move.
Saccade is to look,
Yeah,
and then such that you get your best senses towards the 40 bits that
yeah, exactly.
you
But
put the
tell me
have. Yeah.
what is a saccade, because that's
Or
saccade is
saccade
something.
is your gaze shift. So if
Yeah.
I look straight forward and suddenly I look to the left,
Mm,
your eyeball just did a ballistic jump, and this
mm,
jump technically bad jargon or
yeah.
good jargon
Yeah.
it's called saccade. And so
Mm.
this looking, you actually move your eye 3 times a second.
Exactly. And that's a fascinating thing because it's not something you notice with yourselves,
That
right? You
means
don't,
a lot of it is controlled by involuntary part of your
yeah.
brain.
But also you don't see the jumps. You don't feel that you don't see the like the image moving, right?
That's
You're
right. So
just
your
blind
brain
during the
functions.
saccade. Yeah.
That's right. Your brain, filters out or compensates, and you
Uh,
you do not feel the world is jumping.
yeah.
Yeah,
And
yeah.
you cannot see in the mirror. You cannot see that your saccades, right? Even when
That's
you,
right. There's a little bit of suppressive period. Saccade
yeah.
is roughly about 20
Anyway, so
minutes.
that's sort of
Yeah.
so then you need sort of to okay. So then you have this whatever this thing what you call saliency. You find this thing by looking that this is most important, and then you can sort of move. If you know now that you know where to go, and then you can sort of move your gaze there. But
That's
how does so that is something that the V1 primary visual cortex reports to another brain structure. right?
That's right. This was the hypothesis in the
Mm.
1990s. It was hypothesized in 1990 that was the case, and then the experimental data start to come up and start to show yes, that's the case in 2000s, and now
Yeah,
even
what is the case that is like, because I think you mentioned the superior colliculus as this area that governs, that receives input from V1 and
that's
somehow
right.
governs the eye
Superior
muscles.
corticus is this midbrain area
Mm.
that is receiving the input, then go tell your eye muscle to
Yeah.
move your eyes.
Mm.
And so you can say superior corticus is almost like a printer
Mm.
that received the command from CPU that is
Mm.
V1,
Mm.
and then the printer say, okay, now you tell me that to the left is important to print. So let's go print it.
Mm.
Then tell the muscle to print that, which
Mm.
is move to the
Yeah.
left. Yeah.
And that is still like the best good hypothesis, that it's really superior colliculus doing this, or
Superior colliculus is actually a very ancient structure in almost all vertebrate. Yeah, so
okay.
fish has it, bird have it. We mammals have a neocortex, and in
Mm.
neocortex there's a V1. But
Mm.
for fish, they don't have a neocortex. They don't have V1. So
Mm.
superior corticus have been doing it for all these animals, but the mammals start to grow a neocortex. It appears that the primary visual cortex then start to take over the
Mm.
superior cortex and start take over a lot of the jobs. So then superior become the like executive secretary rather than the boss. As the
Okay.
boss moved to the neocortex, and one of the boss was it becomes the primary visual cortex.
Ah.
That was the hypothesis.
Yeah.
But now it's getting lots of behavior and neural support that indeed is the case.
Okay, cool.
Yeah.
Yeah.
Uh-huh.
So that's something I sort of can remember and sort of
Uh-huh.
understand. So then you have this, yeah, and then and then you sort of, okay. So then we have this mechanism. V1 determine what is most important, the saliency,
Uh-huh.
and then it sends messages or the directions to superior colliculus, and they sort of like send again directions to the eye muscles and
That's right.
point
So
it.
V1 actually assigns a saliency value
Mhm.
to each location of your visual field.
Mhm.
Let's say this location has a saliency value of 100. This another location has a saliency value of 25. This another location
Mhm.
90. So it's a map of saliency value. That means how important
Across.
it is across the whole field. Then
uh,
superior cortex receives this map and then trying to say who is the biggest one. And they
okay.
found, oh, the biggest one is 2 degrees to the left and 5 degrees to the
Mm.
above.
Yeah.
Then they say, okay, it's as if you go to superior cricket for your Olympic final. Okay, it's like
Okay.
V1 got all these uh,
Uh,
local
candidates.
competitions or whatever, and
Yeah.
then the Olympic final. So here's the finalist at this location. And then superior cricket then tells the eye muscle
Yeah.
to
Because it's
move.
only one winner, I guess.
Only because you can only move to one location. Yeah,
Yeah.
your
Exactly.
eye. You cannot do some, not like there's some
It's like,
animals like one eye look at this and one eye look at that.
yeah.
But we humans,
Ah,
we primates, we just
it's,
only one location.
it's only
And so you
one
can say
gold
that.
winner. Mm.
So superior characters, you can say, is the judge about who is the highest.
Yeah.
But the saliency value, how each location have this saliency value is
Mm.
computed in V1. Yeah.
Because this, I think, I mean, this theory that you have developed has sort of been
Yeah.
developed over a few. Well, I think that this idea of V1 as a saliency map, that's sort of like from around year 2000 or roughly.
That's right. So I proposed it actually in the beginning. It was 1997. That
Hmm.
was in a conference,
Yeah.
and then the real publication came out like 2 years later, and then
Ah.
the theoretical publication. But at that time, it was seen as if it's just a speculation because
Mm.
traditionally it's been thought that this attentional guidance is a very complicated behavior.
Mm.
After all, you have to find within the whole book
Mm.
which page to read, which two sentences
Yeah.
to read. It
Mm.
must be very, very complicated. So they think it's the at the frontal area or it's your
Mm.
parietal area, which is more in the front part of your brain
Mm.
is doing it. Well, V1 is the back part of the brain kind of
Mm.
doing very stupid things. And so therefore when I first proposed that theory, it was seen as speculations.
Mm. But yeah,
And
so how was your theory? Your theory was sort of like you just take whatever is known about V1 and make
that's
it
right. So in the
simple. So
beginning,
it's
it
very
was indeed with theory, it was only demonstrated by a circuit simulation of V1. You are very familiar how to do these circuit simulations, yeah? And so I said, okay, let's just do it and demonstrate in the software V1. It looks like so, but still it's only a circuit. Do you have a real data? And so things
But
really,
tell a little bit about what the circuit looks like. I mean, this
our
is
circuit is
in
like
your
these.
model. Tell about your
Yeah.
model.
And so the idea is you imagine V1, you have, actually 100 million neurons, but let's say all these 100 million, each is in charge of different locations of the visual field. They all kind of looking at one little location, but they talk to each other in a way they're connected. And this connection talking to each other as if it's a whole circuit. And so they
So
each
there
of
are
them
all these recurrent connections between these.
recurrent connections between them. It becomes recurrent in the sense that I connect to you, you connect to me. I'm one V1 neuron, you're another V1 neuron. We talk to each other, but they are kind of local neighbors talk to each other. It's not like I'm talking to a very, very remote location. Yeah, not so.
Yeah.
And when they talk to each other, they kind of negotiate with each other in some kind of a way, which then get the most salient location getting much more active than the less salient location. And this activity becomes the saliency kind of a bidding value that you can then download to the superior collectors, and the superior collectors then can say, okay, this one has higher value than that, and I move my muscle, tell the downstream muscle area to move the eye to it. Yeah.
So for example, if I have a collection of like black bars all over the black oriented bars
Yeah.
in some direction and all over the
Yeah.
field, and then you have like one red bar.
That's right.
Yeah. So why then? Of course, everybody knows if you see this image, you'll immediately notice this red bar. It just
That's right.
pops out, right? I mean, it's
It
sort of
pops out. That's right.
extremely
And so
fast.
behaviorally, you feel like, wow, even though
Yeah.
you could not see the gorilla,
Yeah.
but somehow this red bar. So imagine you have
Yeah.
all the bars are black, and there's one red one. You automatically, you don't have to tell people, look there, they will see it.
Exactly. So this will be the clear winner of this saliency race.
Exactly. So the V1 neuron responding to this red bar will be more active than V1 neuron
So
to the
what aspect of a model makes this? I mean, you have all these bars, and there's nothing particular about red. The only thing that the red is isolated, alone.
That's right.
So why is this red bar becoming so salient in your model?
Okay, so in the model, this recurrent connection is such. Imagine V1 neurons. Maybe let's say not color, just to orientation. Imagine all bars are vertical. Okay, imagine you have 10 by 10 array of bars in your visual field. They are all kind of standing up in the array, 10 by 10 array. They're all vertical. But imagine one of this bar is horizontal. Okay, and this horizontal bar will evoke V1 responses much higher than all the 10 by 10, 99 vertical bar. The idea is V1 neuron talk to each other in a way that if you are responding to a vertical bar, you like a vertical bar, and another neuron also like vertical bar, and they're nearby to each other, then they have their recurrent interaction such that they suppress each other. But this suppression is like to like suppression. If I like A, you also like A, then we suppress. If you like B, I like A, then we don't suppress. So therefore, in this neuron that uniquely responding to the horizontal bar is not being suppressed by neurons responding to the vertical bar. And that's why all the vertical bars are suppressing each other, and this horizontal bar is not being suppressed. That's why it's having higher activity. And then the superior collectors see that and say, oh, you are the most salient. I should move my eye to you. Similarly, V1 neurons also tuned, they can respond to color. So imagine all V1 neurons respond to, you have a scene of black bars, so all the neurons respond to black bars suppress each other. And on the neuron responding to the red bar not being suppressed by neurons responding to the black bar, and therefore this uniquely red bar among black bars will give highest V1 response, and therefore,
It's a very elegant and simple, or in some like you don't need much fancy network architecture to do it, did you? And
that's right.
like,
in retrospect, it really is quite simple. The only complication actually came from this neurodynamics of instability.
Okay.
So when you have these recurrent mutual suppressions, you need to make it stable such that you don't have an accidental winner. So imagine you have 10 by 10 array of neurons. They are all responding to vertical bars. They are all vertical. And they suppress each other. If they suppress too much, you can have spontaneous symmetry breaking, which means just by little fluctuation, one neuron
Okay.
somehow become a little bit of a winner, and other neuron become a little bit of loser. Then the winner get get richer, kind of the winner get even more winning. Then you end up having a fake winner. That's a fake saliency. So there is a neurodynamics problem that I needed to solve in order to make sure this does work. So even though in the beginning you feel like, oh, conceptually it seemed to work, yeah, but you say, this dynamics, should I, take care of it? And turns out this taking care of this dynamics was actually the technical problem that need to be solved before you can be certain, at least in the neural network model, that it is feasible. Yeah.
So this was a model problem in some sense.
There was a model problem.
Yeah.
And so in the sense that, if you can't even do it in a model, then perhaps it's not really in a real brain.
That's true.
Yeah. So therefore, it was like, let's see, can you actually do it in a model? And then once you can do it in a model, then you realize, say, hey, well, maybe it's not a coincidence. Maybe the brain is like so. Yeah. Then you're there to propose that indeed.
So, and then, of course, how did you, what is really nice with sort of like, this kind of modeling is that you can actually test it with what's like human psychophysics. So just, psychophysics is, you should just explain what psychophysics is.
So psychophysics is like psychology done in a little bit more rigorous way. So psychology done in an almost physics way. So the idea is a psychology on what you can see, what you can do to a visual input. You measure, like, do you see this or do you not see this? How fast can you see this? How dim you can make an input and still see it? And how fast you can find a red bar among green bars? And this is behavior. So people in psychology have found that, yes, you can find a red bar among black bar very easily. But if you find a red bar among other red bars, a little bit greenish, mixed together, it's difficult. And so a lot of these data have already been available, but they are in a different field, which is a psychology field. While neuroscientists looking into V1 and retinal neurons are not as familiar with these data. And so therefore, in a sense, this does require knowing both field of data to realize that, well, they might be connected. And so therefore, in the beginning, you can say, we can do psychophysics on the software V1 to see if it works. And if it produces the same kind of behavior, like a red bar among green bar is salient, a vertical bar among horizontal bar is salient. These are simple ones. You can also get more complicated ones. And when you find out that, well, the software V1 actually does work like a real psychophysics, then you'll say, well, is this a coincidence or not? Yeah, so maybe not. Yeah.
But how, psychophysics, if you sort of want to measure, then if you show a red bar, like something that pops out, versus something that doesn't pop out, I guess. Sometimes on Facebook you get this sort of see how long time it takes to find the O among the whatever or something, right? And sometimes you just have to look through it sequentially, and it takes forever.
Exactly. So indeed, that's just like
it's not a pop out, right?
That's right. So they measure like how long does it take for you to find a target. And if it takes very short time to find a target, that means it must be salient. It must be because somehow automatically your eye moved towards that location. And that means it must be salient. But if it takes a long time, your eye did not automatically move to that location. You have to scan around on the visual field before you find it. So it must be not salient. So therefore, saliency become a measurable thing through the time it takes. Shorter the better.
But in your model, of course, then it's like how do you measure saliency in a model?
So saliency is measured by, imagine you have n neurons from V1, and one is firing 10 spikes, firing, this is a neuron firing action potential activity level 10, another activity level 9, another activity. And so you say, compare this activity level with all other activity level, and you see how much it's an outlier being a much more winner. So you can use something called Z-score. That means it's relative ratio. You can say, what is the average activity? Oh, it's 5. And what is the standard deviation activity? It's 1. But you are 10, so you are 5 sigma away from that's the idea. So so in a sense that you are all bidding for attention, and the bidding value is 10 and 5 and 6. And the superior character is just the auction bidding judgment and say,
Yeah.
hey, who bids high, I give my attention to who.
But it also superior colliculus reacts faster with this one clear winner, right?
That's right.
Yeah.
That's
So
right.
it's like
Yeah.
one is 10 and the other is 2, right? Then
Something like that.
oh
Yeah.
yeah, exactly. Some
That's right.
kind of so
Yeah.
that's
So
sort of how you. So that's a very elegant. It's actually is a way to link neural firing rates in your model to something you can measure on humans in terms of
that's
this.
right.
Yeah,
That's
that's
right.
really very very nice. It's not too many
Thank
examples of
you.
this.
Yeah, so that was, showing that interdiscipline is beneficial if you can make this cross.
Mm,
Yeah, and so for instance, you can say the z-score higher, then reaction time should be shorter.
mm,
Imagine your z-score is only 0.5. Yeah, maybe you're a winner, but you only win by 1%.
yeah.
Then reaction time will be lower. But if you win by like a 300%, then your reaction time will be really short. Yeah.
Really short. Yeah. Yeah.
Yeah. Yeah.
Cool. So what is the so now we're talking about saliency detection. So what is the role of higher visual areas in the saliency detection? I mean, now you're saying that V1 can maybe do it mainly by itself, but there's something about when you expect
Yeah.
when you look, yeah, exactly, when you have some like clue what to look for.
That's right. So sometimes you can also have a clue what you look for.
Uh,
So superior curriculum is not just listening to V1's input. It also receives input from the frontal eye field, which is in your frontal part of brain, which is normally people think executive function, decision making, very important. And also parietal part of the brain is also trying to direct your attention. They also talk to superior collicus. So you can see superior collicus is an executive secretary with many bosses. Yeah, V1 is just one of these bosses that is fast and very quick, but maybe not so clever. Maybe maybe frontal eye field is very wise but
yeah,
slower. Yeah,
yeah,
and so therefore superior clickers sometimes
have to
can
negotiate.
also listen to.
Yeah.
Yeah, so imagine if you're a primary school student, your teacher says, hey, don't be distracted by the chirping bird outside and listen to me, and I'm giving you a lecture. This is listening to the teacher is like your frontal eye field, frontal part of the brain, telling superior critics, don't listen to V1. Okay, listen to me. So you can use the expectation that my teacher is right in front of me. Just look at her and don't look to
Has
this.
it been like, I mean, of course, this on humans, you don't have this kind of studies, but in some animals,
Yeah,
maybe macaque, then you can measure from superior colliculus. I guess has there been
that's
this
right.
winner takes all kind of experiments that or sorry, has it been like experiments of superior
There
colliculus
have been
showing this?
experiments, yeah. Except experiments are very big. You imagine species has 1 million neurons or 1,000 neurons, then it's really difficult to look at all 1,000. But they can look at, okay, give just two input to the visual field and look at two neurons and see how they
Compete.
compete. But more direct is actually by lesion studies. And
Okay.
what they do is say, hey, how about let's take frontal eye field away, see what happens.
I see.
And then you say, how about we take superior crystal away? What happened? Well, how about we take V1 away? What happened? And people, of course, find that if you take frontal eye field away, the animal monkey wakes up and it can still look around and you get the food to eat and not a problem because but this monkey become more impulsive. Okay, so
Oh.
less controlled. Yeah, because the frontal eye field, the clever, the wise control, top-down control part is missing. Yeah.
Yeah.
But however, if you take V1 away, then the animal can't see anymore, right? And then you say, you're not sure whether V1 is directing attention or it's just that you take the eye away, the animal can't see anymore.
Yeah.
And so that was a very difficult part. It's like you can also take your heart away, the animal will die. They say, oh, attention controlled by your heart. Yeah. So it's very
So
difficult.
yeah.
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 And so, but nevertheless, But if you take V1 away, you find that animal take about 2 months, which is monkeys, not rodents. Takes 2 months, then they can saccade again. But this
Okay.
2 months perhaps requires a little neural rewiring. So retina directly to superior cortex can also guide eye movements.
I see.
It takes 2 months of rewiring. However, if you take frontal eye field away, the monkey wakes up from the surgery, can immediately look around and
I
behave.
see.
Yeah.
So this saliency model is like step one of your grand theory for vision, which is the other one, is this central peripheral dichotomy, which has more to do with like the feedback, right? The second step, what you do after you have sort of how is the higher visual areas, how are that sort of like determining what you see. So tell about that
Yeah. So in the
theory.
sense that, in a sense that you need looking is again in the background that you have attentional bottleneck. And because you have attentional bottleneck, you need to first select this 40 bits. But after the selection is in order to read this 40 bits. And so therefore you say, okay, I just put my central visual field to it in order to read. And so therefore the seeing is to read. And then you say, well, if you selected in V1, then reading should start right away at V2,
Okay.
V3, V4. So therefore you say central visual field is to read, to see, and peripheral visual field is to look, because it tells you to look to the left or to the right, because your peripheral is left and right and top and down tell you which direction to look. And so this is one part of the central peripheral dichotomy is that.
So how does this? Okay, so now we have this central thing that is supposed to get information so that you can see.
Yeah.
But then there are like feed forward and feedback
Feedback.
connections, like
That's
axons
right.
going both ways.
That's right. So the idea is, it's like people say, "Are you sure in peripheral I cannot read?" Well, you
Okay.
can read. It's just poorer.
Okay.
And so they say, well, sometimes it's not as if only central can read. It's just central can read better. Then the question, why is it reading better? Well, it's not only because in the photoreceptors in your retina has more dense spatial resolution in the fovea, but also because you have feedback in the sense that, well, the bottleneck is also in central vision. It's not just in peripheral vision. Yeah. So imagine the bottleneck is so strong that I cannot recognize your face. Then I would say, well, 1 second I only have 40 bits. Maybe I need another 40 bits, or maybe a split second I only get 2 bits. At 2 bits I can't recognize your face. I maybe need another 5 bits before I can tell who you are. So therefore, you kind of need to need the central feedback to fetch the additional bits. And this fetch the additional bit is depending on, do you want to recognize a face or do you want to recognize a tree? Yeah.
Okay.
And so therefore, in a sense, like if top-down feedback is trying to get additional information for the current thing that I'm trying to recognize.
Okay.
And so therefore, because you have central peripheral division in terms of the jobs to do. So central vision is in order to read. Therefore, the feedback should be mainly for central vision, not for peripheral vision. They say, why not? Well, if you put feedback everywhere, the brain will be too big, and we just cannot afford to have such a huge brain. So if you engineer-wise, you have a limited resources, then you put it where it matters. And so that's another part of the hypothesis that feedback is mainly to central vision. And so far, this is still a hypothesis. So let me distinguish between these two theories. One is, say, V1 is a saliency map guide looking. This one experimentally is more and more tested and confirmed. And so, by the way, this is actually the leading theory in terms of falsifiable predictions and how many of them confirmed in visual cortex. So that's
you mean
great,
the latent, the visual V1 latency, no, V1
V1 being the saliency map.
V1 saliency.
V1
Exactly.
being the saliency map may not be the most famous theory because, however, it is the leading theory in terms of how many falsifiable precise predictions are there.
Okay.
The second is how many of these are actually falsified and tested. First of all, 0 falsified. Secondly, multiple confirmed. And so in this sense, this is actually the leading theory. Yeah. And then with that, you say, well, if that's the case, then what's, it inspire you to think about what's next. So the central proof dichotomy so far is still a theory in terms of some predictions come out falsifiable and tested, and others are yet still waiting, especially the critical one about feedback only to central visual field. We have indirect evidence, but not yet direct evidence, but no 0 evidence against it yet. And so therefore, it is still a falsifiable theory, but yet to be more convincingly confirmed than the V1 saliency map theory.
Okay, so the first, so you're saying that one thing that you have more the V1 saliency theory is more you're more sure about that because of how it explains experiments.
That's right.
And then the other theory is sort of like, is it that like roughly speaking, that is sort of, you have this central vision which has a lot of sort of like feedback connections, so that it can go back and forth and see
That's
things.
right.
And then you have this peripheral part which has feed forward connections, but
That's right.
not so much feedback. So in
That's
some sense, the peripheral vision looks a little bit more like what you have in these convolutional neural nets that is so
that's
popular in
right.
AI, right?
Right.
Where there's also little feedback.
Correct.
I think you also mentioned that sort of like these visual illusions
That's right.
that they typically also happen in the periphery.
That's right. And so therefore you say, well, convolutional neural have a weakness, which is security weakness. That means you can hack it. Yeah. And it's, you can cheat it. And this
Okay,
cheating is called adversarial attack. And
exactly.
so you say, well, I know how it's wired, then I know where it's a soft spot, and I can push that button and lie to it. And turns out that in human peripheral vision, you can also do that. And so one of the theoretical and experimental development we have is, in fact, devise these adversarial attack to human peripheral vision.
Yeah.
And so therefore, when you cheat the human peripheral vision, you will see illusions. And that means even though the visual input doesn't have, let's say, a cat there, you illusory see a cat. That is, you just succeeded in attacking this.
Yeah,
And this
tricking.
has attacking, and this indeed has been predicted. So we have already two illusions predicted that way. And you say, how did you predict? Well, you say, if I know the convolution neural network is wired up this way, then I can,
Yeah.
find whatever to cheat this way.
Mm.
And one of part of this network is V1. We actually know how V1 works.
I see.
By knowing how V1 works, you can devise adversarial attack to it, assuming the bottleneck starts right after V1. And so far, we have predicted one illusion is called reverse depth illusion, and second illusion is called flip total illusion, and both of them experimentally tested and confirmed. And that is also encouraging because, so far, to design illusion from theoretical way is still very rare.
So like exactly like in a design and adversarial attack.
That's right, on a human visual system.
And it's also interesting because these illusions are often sort of, you have to fixate somewhere,
Yeah.
and then something strange happened in the periphery, right? That's
That's right.
often
That's
the
right.
case.
To make sure that it's not in your central vision.
Yeah,
If
because
you
if
go
you
to
move your
central vision,
center,
then the illusion disappear. Why
Yeah.
is that? Because central vision has top-down feedback, which you can say it's called visual understanding, and therefore it does not get so vulnerable to such adversarial attacks. Yeah.
So, but do you have do you have a mathematical model for this central peripheral dichotomy?
So far, actually, mostly it's still conceptual. So in a sense that you can say, I have a computational principle, I have a level of algorithm, but I don't have a detailed implementation in a way that I can. So it's not really worth writing down mathematical model yet. The reason is if I write down mathematical model, I will have tons of parameters. And when the parameters so many, then maybe it's easier to do experimental investigation rather than modeling investigation. Yeah. So therefore, yeah. So this is a little bit different from the V1 saliency hypothesis, where I actually use a model just to see the feasibility study. Here, is actually I can directly see whether this illusion works or not in visual psychophysics rather than in the model, and so therefore it's in a sense that's faster to directly test it on humans. Is this actually, do you have this illusion or not? Yeah. And then you say, well, the model does have illusion, will take you, let's say, 2 years to build a model, get this illusion. But visual psychophysics, just one day. Yeah. And then you say, wow, indeed, it is the illusion as you predicted.
So why? I mean, because if you look at this V1 saliency model that you made 20 years ago, so I mean the model is sort of, I mean, just like some excitatory neurons on a sheet and some inhibitory neurons on a sheet and some connections. So what is hard maybe is to find suitable parameters.
That's right. It is hard to find suitable parameters, and that is the challenge. You say, oh, do you want to test it in behavior? Well, people already have data. So this visual search, how fast to find a red bar among black bar, they already have data. So there isn't as much value for me to do this psychophysics test. While here, when you predict the illusion is only just newly predicted. Nobody has data regarding whether this illusion existed. So it's actually faster for you to do a psychology experiment and say, well, illusion indeed tested. I found 10 people and they also see it. And that's faster than building a model.
But it isn't sort of also if you have a model that gives you, because then you really have control of everything and you say, okay, this model is sufficient to explain a phenomenon, right? While if you only do experiments, then you have more interpretations, right?
Yes. In fact, there is such a model already there, but in the sense that it's so straightforward. You can say it's almost like 2 + 2 = 4. So therefore, if you publish paper 2 + 2 = 4, it's already it's a very simple model. But however, a recurrent neural network model is very complex. So therefore, it's kind of worth publishing. But the model really is say, okay, the V1 input. So the model really say, we have all these V1 getting excited by the visual input. And then assuming only this neuron goes to the bottleneck, then you can have a visual decoding stage. Decoding really is like a neural network. And look at this video, a neuron's response and trying to say, is the input A or B? Yeah. And you find that if that's the case, it will be confused whether it's A or B if you have a bottleneck. Just imagine you have a neural network has 100 neurons. Yeah. And these 100 neurons, if you all feed to the next stage, they will say, oh, input is A or input is very clear. But you say 100 neurons, I'm only allow 1 or 2 neurons to feed to the next stage.
Okay.
The next stage say, I can't tell. Only 1 or 2 neurons. It could be B, so you can design in such a way, even though the input is A, if you only feed forward 2 neurons, you can make the cheat and output say it's B. Okay, and this is a very simple model, almost this perception like a very simple model. So therefore, you don't even need to really actually model it. You can just say it and people get it.
Yeah, but sort of like if you had like this, I guess your idea would be if you had like this, say 100 neurons in V1, and then you had like this higher visual area, and then I guess if you want to do a model should then be such that the feed forward things alone could not be tuned to distinguish, but you needed this feedback.
That's right. So maybe I can't actually say it in this. So imagine we already know V1's neurons are tuned to orientations. So imagine you have a V1 neuron tuned to horizontal orientation. Okay, so imagine the horizontal orientation is a Gabor filter. Well, Gabor filter just means you have a horizontal, white bar next to a horizontal black bar. The black bar is on top of the white bar, something like that, vertically shifted to the white bar. And so therefore, this resistive field will be excited by a white bar exactly at this white region. Horizontal, very good. And this neuron will also be excited by a black bar slightly shifted up and very happy. Yeah. So that's good. Now imagine you give an input, which is a black dot exactly in this black bar part and white dot exactly in the white dot part. But they are actually vertically, black is above the white. They're actually vertically aligned. Now this vertically aligned black-white dot pair will excite this V1 neuron tuned to horizontal. Just that this V1 neuron has a horizontal, this is black on top of white, and you have a black dot. And so you just showed an image that is vertically aligned 2 dots, but you just excited a V1 neuron that's tuned to horizontal. Now imagine this V1 neuron is the only one that's feeding forward through the information bottleneck to the next stage. The next stage will say, hey, neuron number 2 is firing a lot, and this neuron number 2 is tuned to horizontal. So it must be a horizontal input. And so therefore, you just cheated the next stage to think this is a horizontal input, even though the input is vertical. However,
the next stage will get the incorrect interpretation.
Exactly. But if you say not only this neuron is feeding forward, but other neurons, they are only looking at one part of this black dot, one part of the white dot. If you all neuron feed forward, you don't get cheated. So therefore, you need a bottleneck as well as knowledge of V1 to predict these illusions. And so by explaining this, you can see that there is actually no need for me to build a model. Just a few words is already explained. So conceptually, it's quite clear. But nevertheless, you can immediately test this on human behavior, and then immediately they indeed see this thing is vertical, but only in the peripheral field. If you put in central field, they don't say it's vertical. They don't say it's they don't say it's horizontal. They say it's vertical.
So what are now you have you sort of you have a lot of sort of like for this new what's it called? What they call it? Central
Central
peripheral CPD,
dichotomy.
central peripheral dichotomy. So
Yeah,
then you have like these different kinds of like predictions for in
yeah,
terms of human psychophysics experiments,
yeah,
and you
yeah.
mentioned some of them, and then you also have some from macaque physiology.
Macaque physiology was actually a test for the V1 saliency hypothesis.
Okay,
Yeah.
I see.
So there we actually find a trial by trial correlation between V1's response
Hmm.
and the monkey saccade to their location. So imagine going back to the situation where you have 10 by 10 vertical bars and one horizontal bar where the monkey is supposed to find and immediately make an eye movement to it. And of course, how fast they make an eye movement depends on how salient that bar is. However, from trial to trial, you find that sometimes the monkey moves eye very quickly, sometimes monkey moves eye very slowly. Then you go measure from V1's activity, you'll find that those trials when the monkey moves eye very quickly are those trials when the neural response to this bar is higher. So there is this correlation. And this is one of the predictions from the V1 saliency hypothesis. However, the central peripheral dichotomy, so far we don't yet have physiological data confirming the prediction, nor any data
At all.
falsifying the prediction. So therefore, it's still a very falsifiable theory.
But you have
But
some kind of an anatomical, like there's something about that, something about from this human this connectomic
that's
studies.
right. So we can have these indirect data. So from Human Connectome Project, from fMRI structural imaging data, you actually have this indirect evidence, which indirect in the sense that you don't know whether these are feed-forward or feedback fibers, or you know is these fiber connected. Or and these are, so in this sense that it already has experimental support. It's just in terms of real anatomical neurons, which is beyond it's much finer resolution. We're still waiting. In fact, I'm actually in a collaboration. Maybe by next summer, we will find it in some. We will get the data to falsify or confirm the prediction. Yeah,
Cool.
yeah,
So I mean, this idea with this back and forth iteration in the central vision.
yeah.
So and that is that could that do you think help in like in this like AI or AI algorithms, because they only feed forward in terms of
That's
the
right.
processing, right? I mean, there's only feedback in terms of learning.
That's
But
right.
this could be like, do you think this could be
That's right.
helpful?
I think so. So in the sense that people always say that can we trust AI? AI says okay, self, driving, turn left, turn right. You say, well, maybe what if it's, and so in a sense that this trust it says as if the AI does not have visual understanding. Okay, you can show it a vegetable. I think it's a gorilla. And that's horrible. And so this understanding, the feedback is actually having a top-down knowledge. So in a sense, you say, well, I know V1 neuron, this neuron is a horizontal tuned neuron. So if it's excited, I know a situation you can excite it by a horizontal vertical pair. And so you actually understand it. If you understand it, you can feedback and say, let's look at another neuron's response
Yeah.
if it's indeed this way. Is it, so this in a sense that this is called generative model. So in a sense that you can generate what should be the V1 response if this is indeed a gorilla, if this is indeed an apple. And when you generate it, then you can go back to V1 and see whether that's the case. And so this is kind of a generative model verification.
Yeah.
And of course, this idea has been kind of around. People in several decades ago, they already talk about, feed forward recognition and feedback generation, generative model. And this, I think, coming from human visual psychophysics, visual cortex like this, hopefully can revive this idea into the artificial neural networks and make it more visual understanding.
Yeah, and maybe like then make it more robust against these adversarial attacks,
That's right. That's right.
like humans are, because we like visual illusions because we are not really threatened by them. We think they're just fun.
Right. That's right. That's right.
Yeah, so
Indeed.
we
Yeah.
always it's always tells something how impression our vision is, that we are sort of we think it's fun when it doesn't work, because
Yes,
it's not a
yes.
problem. It's sort of like it's just like a fun thing
It's
to.
a very fun thing. It really is satisfying to
Yeah,
see, oh, that's why we have this illusion.
yeah,
And how is it under control and not under control? How you can trigger it and how you can not trigger it. And
mm,
that is very satisfying, satisfying our curiosity. Yeah.
yeah. And also sort of like it's sort of like, yeah, you learn about the system when how it works by exploring what happens when it doesn't work in some sense.
That's right. That's right.
Yeah,
It's like it
yeah,
gives us a asymptotic behavior where you can probe.
yeah,
Yeah, you say,
mm,
well, it's like
mm,
when you're trying to understand the law of gravity. The best place to understand it is go to extraterrestrial. Okay,
yeah,
there's no friction, there's no atmosphere.
yeah,
Between the
yeah,
sun and moon, this is how they attract
yeah,
each other. The third planet is far away, it can
yeah,
be negligible or something like that. So we create this artificial situation where it's so simple, it's so asymptote, and you can actually predict very precisely.
mm,
Yeah, and then
mm,
you say, well, it's actually asymptotic behavior. After all, illusions don't happen very often.
mm,
Otherwise, our visual system is in trouble. So therefore, to narrow down these illusions is one of these asymptotic behavior
yeah.
that you have to get to it.
Like these test cases in some sense.
That's right. That's right. So this
Now
is
you sort of actually, like, you just published a paper this year in Trends in Cognitive Science, where you sort of or think about sort of like generalizations of this sort of like center periphery
that's
dichotomy
right.
to other senses. So tell
That's
about that.
right. Yeah. So the idea is this attentional bottleneck, again, is the original idea that you need to, is this attentional bottleneck, if we human have it, then all other animals should have it.
Mm,
Because our human's brain is one of the most powerful. The
mm,
reason we have a bottleneck is because we can't process everything. That's why we have this bottleneck. So other animals also don't have powerful brains, so they should all have bottleneck.
mm,
Therefore, they should all have to select only a small fraction of sensory input to scrutinize. And this selection behavior, in psychology, we call attentional selection. Actually, it's information data science. In informatics, you just select this 40 bits out of 1 megabyte. In some other animal, you will have select this 2 bits out of
mm,
how
mm,
much. Yeah, of course, they already start to go even sampling stage. They are already downsample compared to us, but still they need this selection. And then this says, oh, animal has attention. Well, actually, from informatics, everything has selection. So
mm,
animal orient. They orient by poking their nose here, turning their head there. This is selection. And so therefore this is attentional selection. If you like to use the human analogy, we turn our gaze around is attentional selection. Animal also do that. And of course, not all animals are visual animals. So, for example, if you have a mouse, a mouse maybe is orienting its nose, while we orient our gaze, it's orienting its nose. So we have a central vision. They have their central sense. And their
okay.
central sense is their nose and the whiskers near their nostril. Yeah.
So then they move because birds, for example, they seem to move their heads instead of their eyes.
That's right. Most birds are many birds, especially predatory birds. They are all quite visual.
Mm,
So they move their heads, and then you say, are they moving their beaks or are they
yeah.
moving their eyes? Yeah.
Mm,
Turns out that many birds, their eyes are locked in the heads.
yeah.
So therefore, well, if the
Yeah.
eyes are locked into their heads, are they really moving their head? Yeah. And so, but nevertheless, you can see in their brain, they have a bigger part of the brain to the visual system. Then you know that the vision is the most expensive part.
Mm,
And they probably are moving their eyes. They
mm,
move their heads in order to move their eyes.
mm,
But if you look at a mouse, it also moves its head. Yeah.
yeah.
Well, are they moving their nostrils or are
Yeah.
they moving their eyes? Yeah.
Yeah.
And then you look into their brain, you find that their somatosensory, which is a touch sense by the whiskers, is very big. Their visual system is small, and their smelling system is very big. So you know they're actually moving their snout. Yeah, and so
Okay.
this is how you can link behavior, see what are they orienting with behavior, link into the neuroanatomy and neurophysiology.
Yeah.
Yeah, and
So
so
when I think of when it's sort of like babies, right? When they are like they get a cup or something and they try to chew it from all kinds of directions, right? And they,
that's right. So in a
so it
sense, that you
was
say
like
it's
a
not
gaze. It's sort of like a gaze
That's right. And so
Yeah.
you say it is not absolute. So even in human babies, their eyes haven't quite developed enough.
Uh,
So therefore, their central vision is not as sharp as adult.
mm,
And turns out their sharpest sense is their lips and tongues. So therefore, while you look with your central vision, they try to see using their mouth. So whenever something interesting, they put in their mouth, not
mm,
because they want to eat it, but because they want to examine it.
mm.
However, when they reach the age about 18 months, then their eyes start to catch up. Yeah.
Okay.
Then if by then they still keep on different things, putting things in their mouths, you need to send them to obstetrician, see if they
It's-
see something wrong. Yeah. But before that, it's a normal behavior.
yeah.
And so one of the predictions I actually have on this theory
Yeah.
is if you scan baby's brain, you should see that the brain part devoted to lips and tongues relative to brain part devoted to central vision is bigger compared to adult
Yeah.
human.
Well,
So
that
that's
makes sense.
another falsifiable prediction. Should be not too difficult to do.
No, but then some kind of functional MRI on babies or something.
Something like that, yeah. And so therefore, so in a sense that coming from the physics background where you want to make a theory falsifiable prediction, this is making it very satisfying.
Mm.
So such that whether it's falsified or confirmed is a progress. So it's nice, yeah.
Well, I think has been really nice, and because I think it's really nice to have this theory that is actually making it's like whether it's right or wrong or to what extent it's right or wrong. It's really something that you can sort of it's a clear idea that I can
Thank you.
understand, and then it's something that can be falsified, and then you
Thank
can make progress.
Yeah,
So
yeah.
yeah, so thanks a lot
Thank you.
for coming. Unless you have something you want to add at the end.
Well, I feel like I find it really is an interdisciplinary field. I would like to share this with other people to find that it is such a joyful field that
Mm.
more people can join in, and especially in a climate where people feel like, oh, will we ever find falsifiable brain theories to understand biology? Maybe
Mm.
biology is too complicated. Maybe we will never be as clean as physics. But I would like to say, well, it's just that we haven't got as far as physics yet. But definitely, if we do understand, we should be able to. In fact, we already have examples, for instance, in the V1 saliency hypothesis.
Because I think, I mean, when I sort of look at the field of visual neuroscience,
Mhm.
I mean, that's a huge field. And when you look at sort of like the computational modeling, right, then you have some of these large scale biological network models, like which often then tries to mimic physiology, but
Mhm.
not sort of like human psychophysics. But
Mhm.
you are sort of bridging this in the sense that you have this biophysical, like by at least biophysics motivated model from,
Mhm.
like, for example, the V1 saliency model, where you have these layers of neurons, exciter neurons, inhibitor neurons, and still
Mhm.
comes up with the human psychophysics experiments. But how big is uh, sort of like your sub branch of
Mhm.
visual science? Are there many people working on similar models like you?
There are still few, but if you compare with 20, 30 years ago, it's already quite a big progress. So more
Okay.
and more young people start to appreciate this is the way to go. But it was much more difficult 20, 30 years ago, to say that this is we should, make a progress. So of course, model is actually after the theory. First of all, you have a theory which says what is the functional role of V1. It is
Mm,
trying to guide attention. The
mm,
model is say, well, the best way is you have this theory and you immediately experiment to test it.
mm,
But since I could not immediately test it, then I said, well, can I just test it on a model? Model is like a second kind of you can say assistant to the theory.
okay.
But in the end, even if model tested, it's not a true test. It's kind of a simulated test.
Mm,
And then you say let's test it in real behavior and the neuroscience neural experiment. And this approach, I am very glad to see that more and more young people can go across from theoretical to modeling to
mm,
experiments and back. And I see a future where this is more embraced. And it's part of the reason we have this system neuroscience, system vision science summer school, because there's a demand for it. And it's not just recognized by a few individuals. A few individuals get more individuals. So therefore, we actually have quite a team of faculty members joining to teach in the summer school, and a big response from the young community to
Excellent.
to kind of joining this. Yeah.
So this young community, they are in cognitive science labs typically, or they've experiment, or what part of neuroscience
Yeah,
are
so,
they,
so for
do they
instance,
come
in
from?
this year we have people coming from neuroscience lab and psychology lab, as well as computational labs. So computational labs, they say, okay, can I learn some different models of visual encoding and decoding and attention? And the psychophysics lab and the neuroscience lab, they realize there is a profit to have by being able to collaborate with theoretical people, or they do theory themselves. So one of my postdocs actually became very able to do computational vision. Keith May, he was a pure visual psychophysicist. Then he became such a great modeler that he actually won David Marr medal. So I'm very,
Oh, excellent.
I like to use him as an example that it is possible to go from a psychologist to a computational. Now he's both psychology and computational. But I also see people like computational people become very deep into experimental. So therefore, I see this trend is such a progress compared to, 20, 30 years ago. Imagine if we extrapolate another 20, 30 years, there will be even more. So maybe it will be, hopefully it will be like the traditional,
So
yeah.
you see that like most more visual neuroscience labs have some people doing some modeling.
That's
Is that,
right. That's right. So some of these lab people, the modeling people come, or experimental people come. So we have most students coming from that background for this need. And so in a sense, they say, well, even if I don't become a modeling expert, maybe I can learn enough such that I can collaborate with modelers.
Yeah, no, I think
Yeah,
that's a
or
very,
vice
because
versa.
learning to do modeling, at least like doing, at least we trained a lot, at least I did,
Yeah,
to
yeah,
learn how to do physics type modeling. That took me some years
yeah,
of hard work. So
yeah,
I mean, it's
yeah,
not like something can do. I mean, it's, you
yeah.
cannot do that on the side, but I mean, you can sort of learn to communicate. That's sort of like a
That's
much,
right. That's right. And
that's
so there's
tricky
a
thing.
recognition of such demand,
Yeah.
and people feel that the profit to be earned for young people. They say, well, if I gain another skill that way, now I'm more
Yeah.
competitive in terms of career
Yeah.
development.
But also,
Yes.
I just want to bring. You mentioned something that the theory is more. I mean, like when you say a model or something, that you prefer maybe to like compare theory with experiments. Maybe I misunderstood more directly, but isn't the model a way to make a theory precise? Because it's,
That's right. So, you actually do need. We often use modeling way as well as data analysis, statistical methods to make it precise.
yeah.
So, for instance, let's have an example. You say, hey, if I calculate, law of gravity. Okay, so there's a sun, there's a planet moving around it. You can precisely say what is the trajectory of the,
Okay.
yeah, and then you can measure it, and then you find, wait a minute, somehow this trajectory deviates a little bit from the theoretical prediction.
Yeah.
They say, how could it be? Either Newton's gravity law is falsified,
Mm.
or maybe there's another planet outside pulling.
Yeah.
Okay,
Yeah.
and so therefore you can say, well, only when you can calculate precisely, you can say where that planet is.
Yeah.
Yeah. And then you say, well, okay, 8 o'clock this evening, or 8:05, aim your telescope, this degree and to that part of the sky, you will see a planet moving through. Well, that needs really good theoretical calculation.
Yeah.
Otherwise, it's not quite clear. Otherwise, you may not even notice that this ellipse is actually deviating from the.
And this
Yeah,
story you tell is actually happened in reality, right? With the
yeah, that
discovery
indeed
of
happened in
Neptunium
reality.
or
Means,
Uranus or something. Yeah.
yeah, it was such a triumph. And
Yeah.
so, therefore, for instance, one of the V1 saliency hypothesis experimental tests was actually such a quantitative curve that was predicted with zero parameter. So what is the curve? Curve is the probability distribution of the reaction times in visual search for a unique singleton. So uniquely red bar, uniquely vertical bar. And you can have a probability distribution of the reaction time. And this reaction time distribution is a curve with no parameter of tuning, and that requires quite a lot of actually was not modeling, is kind of statistical data analysis. We can actually write down the equation without a single parameter, and so therefore this equation has 7 quantities. You measure 6 quantity, you can predict the 7th one by this equation, and then you can go experimentally measure these 7 quantities using the 6th measure to predict the 7th one,
Yeah.
see if it matches experimentally with 0 parameter. And this is one of the rare examples where these mathematical techniques, it was used a curve at the whole curve. That was so fantastic. And each quantity is a curve. So therefore, we measure 7 curves, use 6 curves to predict the 7th one, and compare with the experimentally measured 7th one. And they match on top of each other. They match, of course, using the statistical test with p-values and all this. They are not as good match as this, Cobalt telescope of this, black body radiation.
No,
I'm in the Max Planck Institute.
I
You have
think
this
that.
black body radiation because they're really on top of each other. Okay,
Yeah.
the error bar is so short. Okay,
Yeah.
very tiny on top of each
Yeah.
other. And we are also on top of it, but our error bar is a bit longer.
No, that's true.
But nevertheless, it's a 0 parameter.
I
So
think vision is more complicated than the black body or the black body
that's
radiation.
right. So therefore, we still have quite a distance to go to reach that far. But qualitatively, we have already a few examples in computational neuroscience where this is actually reached.
So where should people who are interested in learning more about this? Where should they go? I mean, you have this book that covers quite a bit,
That's right.
and you have of course, the web page, and you have
I have a web page, and there's also the online courses. They can just Google my name, find it. But there's also general background education into the theoretical neuroscience if they want, in a similar spirit. And so there's a textbook by various people, including one Peter Dayan and Larry Abbott, and other people also written maybe even easier level of computational neuroscience. They can be also used for the undergrad level. So people are recognizing this, that they try to kind of push towards the undergrad teaching. And so there's, I feel like I wish I can be the future generation, but nevertheless, the future generation is having more of such opportunities. Yeah.
Excellent. Okay.
Thank
Thanks a
you.
lot.
Thank you.
Okay.
Yeah.